All right, so then if we go into the chloroplast, and we want to focus specifically on the light reactions. Again, this is taking place across the thylakoid membrane, and this is the process that requires light. So these are also called the light-dependent reactions. And so here you'll see I have two thylakoids stacked together. And here, I like this picture because now you can actually put this into perspective. So this whole thing is a chloroplast within the leaf of a plant. And then you have this single thylakoid, and you can see the membrane. And now we can start to visualize what's happening here with the light reactions. And so remember that this is an electron transport chain, which means that we are going to be using electrons to do work for us. And so light is key to this process. So we have light coming in, and then we're also going to use water and harness some electrons from this to help us. So in this process, in order to get these electrons to do work for us, we have to excite them to a higher orbital. And when we do that, they are less stable, but they have more potential energy for us to use. So when this light strikes this photosystem, and remember that photosystem 2 comes before photosystem 1. It seems backwards, but it's about which one was found first. So they discovered photosystem 1 first and gave it the number 1, and then they discovered photosystem 2. But so like I was saying, you have light coming in. That light is going to excite electrons. And when that light excites electrons, we can split water. We're going to split that into oxygen and hydrogen ions. Those hydrogen ions will go on to do some work for us. But then we also can continue to use these excited electrons. So our excited electrons are going to pass through other proteins in the thylakoid membrane. When they reach the cytochrome complex, this is so important to us because it's going to create a concentration gradient. It's going to pump hydrogen ions. I should probably label that here. We're going to be pumping hydrogen ions across this membrane from the stroma, or this outside space, into the thylakoid space of the lumen, across the thylakoid membrane. So now we have more hydrogen ions in the thylakoid space than we do in the stroma. So we have a natural gradient where these hydrogen ions are going to want to move from high to low. That's very nice for us because then we have this ATP synthase hanging out also across that thylakoid membrane, that when those hydrogen ions pass through the ATP synthase, they are going to turn ADP, an inorganic phosphate, into ATP. Now this is usable energy that can go on to the Calvin cycle. But so the electrons don't stop there, okay? So they're not done going through this electron transport chain. They're going to continue on to the first photosystem. And here we take in some more light. So you see this photon of light coming in here because our electrons have done a lot of work and they don't quite have enough energy to do what we need them to do here. So we're going to excite our electrons using the light, and then they're going to pass through other proteins into NADP plus reductase. So reductase should tell you that in this process we are going to reduce NADP plus. And we're going to reduce NADP plus into NADPH. What does that mean again? Remember that when something becomes reduced, it's gaining electrons. So we're reducing the charge on it, we're making it more negative, and we're also going to add a hydrogen ion. So when we add this hydrogen to NADP+, we're also adding 2 electrons. Now this molecule has a lot of potential energy because those electrons can go on and do work for us. So by this point, now we have reached the end of the electron transport chain, and so we have some NADPH being made through this process, and we also have ATP being made. Hopefully this helps you see the 2 reactants coming in and the 2 products coming out, and then all of this in between is just showing us how those electrons are doing work and how we can get to these intermediates from the initial reactants.